From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and people like you who are making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melittologist, Associate Professor of Pollinator Health in the Department of Horticulture. Listeners of Pollination, you are so lucky this month because for the next 3 weeks, you're going to have a different host every week. Starting this week with Marek Stanton, who has a fascinating conversation on the intersections of science and art with Marek Stanton and Jasna Guy. Now, Marek is Kalika's son and has some notoriety in Oregon because he's the designer of our state license plate, or B plate, but also the youngest master melittologist here in Oregon in our bee biodiversity survey. He is talking with Jasna Guy, who is a Canadian multimedia artist who has been working, and many people around the region know Jasna. Maybe you've seen her show in Penticton or at the High Desert Museum in Bend. The conversation is about Jasna and her body of work, but also about a collaboration between Jasna and Marek, which is going to be featured at the Bee Biodiversity Museum in Vancouver, British Columbia, University of British Columbia, opening Thursday, November 16th. So in this episode, we get to hear the back and forth between a young and established artist thinking about really deeply about pollinators and how to kind of get across in an artistic sense, separate from sort of the kind of work that we typically do around the scientific cataloging of biodiversity and our understanding of ecology, how to get this across in aesthetic terms. So this week on Pollination, I'm really looking forward to hearing this conversation between Marek and Jasna.
Jasna, welcome to Pollination. Right. This is Marek Stanton. I'm also known as Marek's mom. And I have the pleasure today of facilitating a conversation with Jasna Guy and Marek Stanton from Jasna's lovely home in British Columbia. Over the past 2 years, these kindred souls have developed a friendship rooted in a love of art and pollination ecology, where Jasna has been able to serve as both a mentor and friend. So, Jasna, I want to first ask you to talk a little bit about your art, which is where Marek and I first came across you at the High Desert Museum in Bend in 2021.
Okay. Hi, Kalika, and hi, Marek. Nice to see both of you. So, the work that I do is two-dimensional, and it's all based upon it's based upon botanical studies and research. My practice is very heavily rooted in research, and then from there I'll kind of extrapolate things that I think might be of interest visually. And the concept behind all of them is to show things to make visible things that are invisible. So, unless you have a microscope on hand all the time, oftentimes you're not going to see what you might be able to see. So a lot of the studies of the flowers are really about discovering wonderful, incredible structures and the various pollen that these plants have, and also the nectar. And one of the things that I love to do is literally taste. So I'll take apart a flower and I'll literally taste, and sometimes I forget that I have to check to see if the flower is toxic or not. And I kind of do it afterwards and I think, oops, that maybe was not a smart thing to do. Anyway, so I can't think of anything better than sitting in a meadow amidst the flowers, inhaling the fragrance of the flowers, listening to the buzz of bees, and tasting nectar. And then also collecting pollen, because that's another critical area of resource for pollinators. So just to get back to the art, yes, I try to be very aesthetically based for the two-dimensional work, because I think that has a kind of attractiveness to it, right? And I collaborate with one of the most wonderful taxonomists in the world, Lincoln Best, to create these. They're kind of environments. Let's put it that way. They're environment. So they kind of mirror each other, because you've got the actual specimens and the bees, but on the wall you have something which is interpreted in a different way, in a two-dimensional way. I also do little quirky animations, which kind of displays a certain aspect of a flower or of a little process, and to me that's just another way of representing all of the work that's there. And I guess the thing is that I like different modes of representation, so that's why you'll have some things that are like the videos. Then there'll be drawings, there'll be things that are mixed, there's the dissections, and then the scans and the photography. So there's a variety of ways of approaching the botanical specimens. So I'm not a botanical artist, but the botanicals are my subject, if I put it that way.
That does work.
Yeah.
Makes sense. Before I move to the next question, Marek, I was wondering, because this is where you first saw Jasna's art, what stood out to you and grabbed your attention?
Well, when I first went into her exhibit in Bend, which is when I saw, when you first entered the room, there's all these walls. So in the middle, you had the specimens of the flowers and the bees, but on the wall, you don't see when you first enter, is all these beautiful little squares of color. And then when you go up closer, you see on them, each has a label of a type of flower. And so you're looking at it and you think, I thought each type of pollen had one color, like only a range of like yellows, maybe some white, orange, maybe some had red, but there's blacks and blues and greens, and not just like a slight pastel color. It's like, it's very vibrant. And then up against the walls, there's all these beautiful pieces of artwork. And of course, it's not just a flower, it's a flower with hidden messages in it. And then there's like little wordings and quotes of famous and other incredible researchers or other artists. And so it's all these hidden intricacies that I really liked.
Well, thank you, Marek. That's very, very cool. I'm glad you caught that. I know you're very careful in your observations, and that you go through everything kind of with a fine-tooth comb when you're studying. So if I might follow up, that Marek then sent me this wonderful long email with at least 3,000 questions attached to it. And I guess I don't know whether he was wondering if I was going to respond or not, but I did, because number one, I was very impressed by his email and the kinds of questions that he gave me. And he also said, well, I've taken this pollen and it doesn't look at all like yours. So anyway, and I said, well, why is that? And so I answered him. I said that the pollens might change, the color might change depending upon the environment in which it is growing, for one thing. And whether it was collected by a bee that mixed it with nectar, that would make another difference. And again, we come up with this issue of, is there dust? Is there dust there? So is the pollen being contaminated with the dust?
And as we looked, we've also explored black lights and how things look so different on black light. And so there might even be like microplastics and other things in there, all sorts of other weird and also maybe slightly disturbing particles.
Yes, absolutely. And that's another aspect of it that we explored, was the UV reflectance and absorption of different parts of flowers. And we didn't realize initially that they all reflect. I mean, they all have the UV chemicals in them, and that the pollinators are able to see them. And that was a very cool and exciting discovery.
Yes.
To learn about that. And actually, that kind of brings me a little bit to one of the wonderful people I had the honor of mentoring, Svea Brusland, who just finished her honors double major at OSU, and she did her study on how bees see flowers and their environment. And so it wasn't just UV, but also bees don't see reds. So you have to take out the reds. So what bees see is totally different from what we think they see. So anyway, that was a very interesting study and that she did.
And also visually appealing. It was a very pretty weather photography that showed that representation in some form that we could actually see ourselves.
Yeah.
So was this another exhibition that you...
Well, this was Svea, Dr. Gail Angelotto brought her, so she brought the students to see the show. And Svea was one of the people, one of the students that saw the show. And she decided that she wanted to apply for the fellowship, which is an art and science fellowship through OSU. And so she did that, and she asked me if I would be her mentor. She needed two. She needed a science mentor, who was Dr. Gail Angelotto, and I was her art mentor. And so we spent a year with Svea, kind of talking and going through what she was working on, which was really it was a wonderful, enriching experience. And I just love this idea of the mix of the art and the science. And then one of the things that's, that is very important, and the things that I struggle with also, is that it isn't sufficient just to, you have a beautiful photo. That doesn't make it a piece of art. It's not necessarily art. It might be a beautiful photo, but it isn't enough. It has to somehow interpret and be translated and give us something more than what's there already, for it to be, I think, an engaging piece of art. So, and when I think of Svea's work that she did at the little gallery, I think that that's what she did, that she was successful in that. And as a matter of fact, she was one of the 3 students chosen to be in a show in Portland next year. A curator came from the Reser Center and chose 3 students, and Svea was one of them.
Oh, that's fantastic.
Yeah. So, one of the people who is instrumental in all of this is Dr. Jerry Bartholomew, in terms of the art and science program at OSU. And they're going to continue it and expand it, and because I think it's been a very positive experience.
Is that the fellowship of...
Yeah.
For art and science?
Yeah, exactly. So I think that's going to be a very positive addition to the department. Yeah.
So you mentored Svea through a partnership of research and art.
Yes.
And from what I've seen in your exhibitions, or just in any of your artwork, that you also naturally come through your art with a marriage to research.
Yes.
Like when I first saw the exhibit in Bend, my first thought was like, this is beautiful. It's visually appealing. There's colors and shapes, and at a closer look, as Marek was saying also, there's words and there's learning and education, both in language as well as in pollination, and about bees and botany and color and types of art.
Yeah.
Can you tell me a little bit more about how you intertwine your art and research? And it seems that in some ways your research is manifested through your art, or is it that you come to art and you look for research to fuel your art, or is it just kind of like chicken and the egg? Like
I think, I think it's sometimes it's one and sometimes it's the other. The research is definitely, it's fundamental to my art practice. So if I didn't have that research, I would not have anything to work with. I mean, I love botanical drawings and illustration, but it isn't sufficient for me to have just that. There has to be kind of a concept, a theme, before I'll undertake it. And for me, that has always been searching for what do flowers contain, for example. So are they sources of nectar for pollinators? Are they is the pollen itself something that the bees require? Is it a place of habitat for them? So what functions does that specific plant play in a pollinator's life? So that's how I approach things. And I will definitely, if I can, I will collect specimens. I am very cognizant of collecting ethically. And excuse me, I'll press the specimens. And so I always have available the specimens. And again, for me, that's another mode of representation, is having the actual pressed specimen. So, and when Marek sent me the email, he had questions on there, and it seemed obvious to me that he was very interested in this kind of work as well, and that I think that there was something that we could share, despite the fact that we were, maybe despite is the wrong word. We're like decades apart. I'm at least 3,000 years older than he is. And so
I would divide that by a couple decades, but
yeah. So anyway, and that was one of the wonderful aspects of it, because Marek approached things from the entomological perspective. He's very much into the various pollinators and especially the bees. And for me, I approached it through the botanicals. And so I found that the two of us together would have interesting conversations in terms of how the bees and pollinators interact with their environment. And I guess that's kind of the bottom line, right? Is what
different
happens.
forms of looking at pollination ecology.
Yeah.
One, I look more at the bees and why do the bees want to visit this flower?
Yeah.
Yeah, it's more why are the flowers, how do they offer things for the bees to come visit them?
Yeah.
How do they get their pollination? And I'm how do the bees get their food source of pollen and nectar?
Yes, exactly. So that's kind of fueled our emails and stuff for the past year, and actually the year before, the same thing. And we're never at a loss, because this is a work in progress. It's we're always looking for new flowers and also new studies. We tried figuring out how many female Umlaria bloom before the males, and how many are there? And then we tried.
What we found was it's just hard to find females. There's more males than females.
And then we tried to collect nectar with those crazy little glass vials. And that was another whole experience for us.
To figure out if it is even nectar that we're collecting, or if it's just morning dew.
Yes, I do remember at least that episode, because we had teeny tiny needle-sized vials sitting around our house, and Marek getting up in the morning or going out throughout the day trying to capture levels
Something.
of nectar to measure throughout the day to compare.
Yeah. How is it? So does it produce, nectar in the morning? Does it produce in the afternoon? How many times a day does it produce its nectar and stuff like that? So we've undertaken a variety of things that kind of nobody had interest in, except us.
And made beautiful pieces of
art as a result.
Yes.
Yes, from as mentioned, I have lots of questions, but of course, from each one question, we have 20 more questions.
Yes, exactly. That was the other thing. It's one question always. Talk about bird walking. Oh my gosh, just everywhere.
So, moving back a step.
Yes.
How did you come to have a focus or an interest in pollination ecology? And for you, that's more plants, but you've also incorporated bees.
Yeah. I have, absolutely. And I actually came to pollination through honeybees. During the first decade of 2000, colony collapse disorder was very much in the news. And, kind of reading all what's been happening, and then I started finding books on the topic and stuff, and various articles in peer-reviewed journals, and I realized that was really quite interesting. And I thought, when you think about a hive, for example, the concept of there being 30,000 to 50,000 bees in a hive, that's incredible. How do creatures live like that. And I thought, okay, well, I'm gonna see if I can represent 50,000 bees. So I went through a variety of ways of doing that. And it actually resulted in an exhibition at the Richmond Art Gallery for that. And so that was a tremendous amount of fun. And then after that, it just kind of expanded. One of the things that happened while I was working on that exhibit was that I realized that it wasn't the honeybees that were in trouble. It was our native bees that were in trouble. And I thought, oh my gosh, so my focus has been incorrect. So I had to reroute it so that it would include native pollinators as well. So that's how that piece actually came into kind of existence in the final analysis, realizing that our native pollinators were really were in trouble. And at the same time, the other thing that I noticed was that pollen was such an important aspect for the bees, crucial for, for queen bees, for the development of their ovaries, crucial for the babies, crucial for the existence of the next generation of bees. So that was very important. And the other thing that I noticed in terms of the pollen, as Marek mentioned, there's all these colors. And that was the case. I thought pollen was yellow. I had no idea that it came in so many different colors. And that was the exciting thing for me, to discover that. And I thought, okay, well, I have to test every single flower on this earth. And there are only over 250,000 angiosperms, I think. Or more.
Only that.
Yeah, only that, yeah. So I've done about 200, so I'm getting there. In the next 50,000 years. Anyway, so that's how I initially started. And when I realized that it was the native pollinators that were so important, just at that time, it's interesting what the universe can do. Along comes this incredible taxonomist, Lincoln Best, and he does this wonderful 5-day workshop and BID class. And this was in 2016, and that was in Kamloops. So that's where I met Lincoln, the real Lincoln. And I think Anthony was there. I think he showed up for that class as well. I don't know whether he stayed the whole time, but he was there. And so that was wonderful. It was amazing. I understood nothing because it was so hard and so laden with,
I think it's improved since then, but maybe not by a huge amount. It's still a lot of information.
Yeah, it is. And for the first time,
vocabulary.
It's always, and vocabulary. Yes,
it's a foreign language.
It is.
It's definitely a foreign language. It was quite amazing, but it was a wonderful course. I appreciated taking it. And then the next time I took the course, which of course I would do, whenever he offered a course, it made more sense. It started making much more sense. And I mean, you've done that, Marek.
I've done it 3 years now, but each year it's still like, oh, well, that seems much easier now.
Yeah.
When what the year previous was a very difficult key or something else to work through, just because of vocabulary. Each year you kind of build on the previous year.
Yeah.
It's a little bit easier, just a little bit.
Yeah. Well, it was through that, after we took that course that I was offered an exhibition at the Seymour Art Gallery here in North Vancouver. And so I asked Lincoln if he'd like to collaborate with me. And that was, the show was called Pressed for Time, because we had pressed specimens, we had mounted specimens, we had all this kind of... Anyway, the title kind of related to everything, because they are pressed for time, right? Absolutely. Their phenology is short for bees and for flowers, right?
Yeah, it's not always necessarily matching up.
Yeah, exactly. And so it seemed an appropriate thing to call the show that. And so Lincoln said yes, and that was our first collaboration, which was wonderful. And then we continued on. We had a second collaboration at the Penticton Art Gallery. I think that was 2019. And Lincoln put together beautiful exhibits of specimens floral specimens and bee specimens for that. And again, we had stuff on the walls. And for that show, I also was able to do some drawing directly on the wall, which was a lot of fun. It was great. So that was very cool. And then we did a third one, and that was Bend. And so his display cases were really gorgeous there as well. So that was lovely. I really appreciated being able to share that, and I was glad that Lincoln wanted to share that kind of space for opening up conversation to people, visitors who would come to the gallery. And as a matter of fact, prime example here is Marek, who came to see the show, and as a result, we've been able to continue our relationship and our research and our study and our love of bees and pollinators and plants. I mean, and art, right?
Art, yes.
Yeah, because we've just continued to explore different aspects of art as well, not just that. So
all kinds of modes and mediums.
Yeah, exactly.
Great. So, I understand that you all have a new exhibition coming up now that COVID is past, and it's going to be at the Beatty Museum of Biodiversity at UBC.
That's right. Yeah, it's not going to be a very large show. I've had some issues with my health, and therefore I just had to kind of cut back almost everything that I do as a result. But I was so pleased to have the opportunity to show there. I had given up actually a big show, opportunity to show there a couple of years ago because of health reasons. But Linda Lipson and Derek Tan were very graciously offered me a space. And so for this show, again, there'll be two-dimensional work, but this show will also involve a collaboration with Marek. We're going to do an animation together, which I think will be very, I think it'll be of course, it'll be on topic, important topic of today. So, Marek, would you like to speak a little bit about your experience so far?
So far, it's been very much a learning experience as I haven't worked with animation before in this mode.
Yeah.
Especially not with Photoshop. So most of my artwork has just been like textual, like physical works that I can like as painting or drawing. So working with Photoshop is definitely just like learning a whole new tool and then doing that, and then also figuring out how you need to draw or paint to be able to transform that into an animation is very interesting, to say the least. But yeah, we're hoping to represent some of the little snippets of different bee life cycles and also point to some of our current issues they are facing.
Related to their, yeah.
Yes.
Mm-hmm. So we're hoping this is going to be highly successful, and it'll be up for the Grammys or the Oscars. So that's where we're aiming, right?
If not national.
Okay.
So with this collaboration, what you're using Photoshop and you're focusing on bees or plants,
pollinators,
or pollinators in general.
Yeah.
And
would you like to explain?
It's like I think the word's like vignettes or something, just like very short little blurbs of snippets of bees' life cycles for 3 species in particular. And then at the end, we have a little symposium of different pollinators that aren't just bees, talking about current threats to them, whether it's like light pollution or more climate change focused ones such as like flooding and or lack of water. Those are extreme droughts, which then can also lead to wildfires. And then as I mentioned a little bit earlier, how that affects phenology of flowers and when they're blooming, and also when bees emerge.
Is there something there for them to eat? Right?
Yeah. Or if there isn't, then how that affects them.
That affects them, exactly. Yeah. So, it's been very interesting, and generally I do the animations on my own, but we've really shared everything here. There isn't so far there isn't a single thing that doesn't have both our hands in it. You know, when we talk about absolutely everything, including down to the words and letters and everything. So it is as much a collaboration as we could possibly make it, I think in terms of this. So for both of us, I think it's a really cool experience. And I really do love the fact that we are, you know, we're it's an intergenerational kind of project and undertaking, which is so cool, right?
Yes.
It's
different perspectives and experiences. Both like growing up into this new thing, but then also having the ability to be able to see these kind of changes happen.
Yes.
And know what it was before a lot of that had happened as of late.
Yeah, exactly.
So to give a little bit of background, Marek has been in BC with Jasna working on this for the last week. But this is a big project, so you're not finished with it.
Definitely not.
Yet.
We're going to do this the long distance way. Yeah. So, you know, because it's digital, we can scan. Marek will scan his drawings, I will scan my drawings, and then we'll put it together. And I think that what's going to happen at the end is that we'll get a professional animator to put all of the work together for us so that it's smooth and seamless. Just because in working with Photoshop, there's a limit to how much you can do at one time. At least that's what I found. And maybe I just don't understand it well enough, which is possible. And so anyway, yeah. And I mean, the Photoshop is lovely. The timeline function is great, but at the same time, it gives you the opportunity to do short things. And that's what I've been doing mostly. And this is going to be longer. So I think we're going to need to involve someone at the end of that process to help us out. Yeah.
So you've got your work cut out for you. And
Definitely.
when will this exhibition be opening?
Well, I think it's going to open mid-November. And theoretically, it might go on for several months. I'm not 100% sure, so please don't quote me on that. I'm going to find out very, very soon what the dates are for the exhibit. So, yeah.
Well, super. I look forward to seeing how this project progresses as well as the final product.
Yeah.
I do too.
Me too. Yeah.
That's part of the creative part of it, is
Yes.
flexibility, whereas science is often more black and white.
Yeah.
But taking research as well as art, you don't always have a definite end point.
Yes, exactly. And that's one of the things that I think is very important to understand with any art making, is that it is a process, and you things change as a result of that process. So it's very hard just to say, okay, this is what this is going to be. I'm going to have these images and that's it. It just doesn't work that way. You know, you might start with those images, but inevitably everything kind of goes in all sorts of directions, and you have to be willing to go with it in order to, you know, it's learning doing that. It's absolute learning, and it's great because the more you do that, the more creativity comes out of it. At least that's what I found.
Has that been your experience with each of your exhibitions?
Yes, always.
Okay.
Always. It's always a matter of, well, you know, you want the exhibition to be related to the place in which it is, because that makes more sense. So, you know, if I'm doing the botanicals, I'm using flowers that are within that area. So, you know, for Oregon, I did flowers that we share with Oregon. In Washington, it was for flowers that we shared with Washington. So, and for the BT, I'm thinking, of course, I mean, the flowers that I chose are flowers that we have here. But the other aspect is that BT is a science museum. And so they are very heavy obviously, into their vocabulary, whether it's entomological or botanical. So I'm kind of exploring that aspect of it, of the museum as well. Is there ice cream after this?
Fantastic question. It is your home. Is there ice cream after this? I'm hoping for yes.
Have to look in the freezer.
I want to at least express thankfulness to you and your openness to educating, Marek in particular, but the general public at large, that your exhibitions are beautiful.
Thank you.
and educational in a very dynamic way that's engaging.
Thank you.
More so than a textbook or just a science exhibit.
Mm-hmm.
That you can have the marriage of these two different parts of your brain is approachable for a lot of people in a way that maybe isn't otherwise. That science can be beautiful.
Yeah.
But I've loved watching this process and the development, the learning that goes both ways, the questions and the research and
It's been wonderful for me. It's been, to be with Marek has been an extraordinary experience, I must admit, because to find a young person who is so interested and dedicated to not just ecology, but to a subject, whether it's the art or the science aspect of it, I think is very rare. And I really appreciated the fact that he was interested in the art and, what makes for the art, why the art, and where, what role does the science play in your work, and how do you put them together, and why is this important, and, and why is this not important? It's like, what makes for an interesting piece of work, and why is that not an interesting piece of work? Because those are questions that he would ask as well. Is this art? Is this, or what is this? So, for example, you've got sometimes you can have an artist who will come and will help you show things in science, so develop visuals. But that doesn't necessarily make it an art exhibit. It is an artist helping science. But then you have art and science, which is kind of, it's based upon research, but it wants to be, its end product is aesthetic. And then as Jerry Bartholomew noted just recently, then there's the third one where this is a very dynamic one, where an artist and a scientist get together and they create something from their mutual kinds of work. And I think that's really, really very exciting. So from what I was reading from her recent missive was the fact that there are three forms of interrelationship, engagement in terms of art and science. And I think Marek and I fall into the second one because we both love the science, but we both love the art. So we're not, I'm certainly not a scientist. He might wind up being one.
We'll see.
Right? Yeah. But with a very strong bent for art. And so, I think that's where we fall in terms of where are we in terms of art and science.
Would you agree with that, Marek? Where do you see yourself as far as science and art?
I would say it's hard to say one or other. It's like I've had one hand in both as long as I can remember. It's always been the love of animals and the natural world, but on the other side, also always a love of art. So it's not really like ever been separated from me. It's always been together. The one same for what I was interested in drawing or representing was always involving the sciences in the natural world. Yeah.
Yep. It's true from everything that I've seen of your work. Definitely.
Lots of animals and plants. Not many people.
Yeah.
interest levels.
Yeah.
They're not the top.
Yeah. People are interesting, but bees are more so. I love their buzz.
Yep.
We have, let's see. Though I'm not normally hosting or interviewing, my understanding is we have 3 questions that need to be answered.
Okay.
What is your favorite tool? What's your favorite pollinator? And do you have a book recommendation?
Okay. Interesting.
Who would like to start?
How about you, Marek? Would you like to start?
Sure. Could we go with one?
Sure.
and
How
back and forth
about you start
each?
with our favorite
Sure.
tool?
I think my favorite tool are my hands because I can use them to do all sorts of different things. They're not set to just one task. I can do it for multiple different things, whether that's making my tea in the morning, or that's drawing something in the afternoon, or otherwise.
Fantastic.
That's great. That's wonderful. So I'm going to go to the opposite end of that, and I'm going to go to the computer, and I'm going to say that the function in Photoshop that allows me to create an animation is the tool that I really like the best. So at the moment.
I can agree with that, though. It's a very cool tool because then you can see the little animation play out before you. It's very fun.
Yeah. So.
Well, fantastic. How about favorite pollinator?
Yes, no, you can go first.
Thank you. I find that really hard, but I'm going to say that I'm going to say hummingbirds, and I'm going to say the Anna's hummingbird because it's around here all year, although I would actually pick the rufus that still migrates. But it's the hummingbird. I'm just fascinated by the fact that a tiny bird like this can be so important to our environment. So, yeah, that's why.
Thank you.
Mm-hmm.
Merrick,
I
favorite pollinator?
Say I really like all the really pretty or weird looking bees, like the ones with all the weird features or bright metallic blue or green. But my favorite bee, at least the one I've generally liked the most, is called Halictus legatus, and it is a very boring bee. It's very boring in how it looks. And most people would be like, oh, okay, you like that one. I'm like, yep. And it's kind of like a dull metallic. You don't really, they're very common. It's not even rare by any means. It's found throughout the entirety of the US.
Okay, so Merrick, can you sell us on why this is your favorite pollinator?
Yeah, that's exactly what I was going to ask. That was going to be my question.
But the thing that I like about this and that's so unique about it is that unlike many other pollinators, because most of our pollinators are solitary, that this bee goes from highly social, like a bumblebee, when it has like a queen and workers,
Really?
to the exact opposite end where it'll be solitary. And then it has everything in between, having cooperative females that are splitting up tasks and they're just like sisters, or one female that is kind of like, okay, now I'm the one in charge and you guys are just gonna obey me because I'm bigger than you. And then the other ones are just like, okay, I'm going to listen now because I don't want to be chopped into two or stung. So they have this very cool range of social behaviors. So I find them cool because they have this cool, more cognitive and behavior things that you don't necessarily see just on the surface.
Wow, so does that mean that within the species itself?
Yeah, so like depending on where you find it, will depend on kind of how social it is.
Really? So is it something to do with the soil environment, or have you been able to discern?
From what I've read, it's partly where they're found. Like if it's on a mountain, because mountains are like higher latitudinally, it'll have like a shorter season. So they don't have as much time to be able to have the worker caste and then to have the reproductive caste.
Yeah.
So they just go straight to having the reproductive as in like they're solitary.
Interesting.
If they have a longer period, they can coerce their children into being their slaves in a sense. But it does have a positive benefit for their children because they're more closely related to their siblings than to their mom, or even sometimes their own offspring. So that's partly why eusocial and other behaviors arise. It's very interesting.
Yeah, no,
at least for me.
That is very interesting. So tell me something. What happens to the males? In those situations?
Males are very dull anyway, right?
Yeah.
But in this case, it's kind of like bumblebees, and they're just there for the mating at the very end, so the females can get their eggs fertilized, and then they're done.
It.
That's like
same thing.
what they do. I mean, they do pollinate, but not quite as great of pollinators as like the females, of course, because
Right.
they're not
'Cause
collecting
they're
the
not
pollen.
collecting.
It's
Yeah.
more incidental.
Yeah.
Like I think they will still eat the pollen, but it's not necessarily like they're
Mhm.
I'm going to take this pollen from plant A to plant B, like the females are.
Yeah.
So,
Gotcha.
yep.
Okay.
Thank you for explaining that, because
So
that's
they are
really
interesting.
interesting.
Yeah.
Just not the prettiest.
All right. And the last question then is for a book recommendation. Do you have a book recommendation? Merrick, you get to go first.
Okay. I would say a book that both of us have really enjoyed is called The Boy, the Mole, the Fox, and the Horse. And it's a very pretty book with wonderful quotes that are both wise and also gorgeous artwork. And it's by Charlie Mackenzie. And he is, I think, British. I'm not sure. But
And they
quite
made this
enjoyed it.
great animation.
They did
of it.
too. They also made an award-winning film,
Yeah, they did.
which is quite beautiful as well. And it's almost just like a movement version of the book.
Yeah, it is.
What about you, Jasna?
Well, you know what? I found it very difficult to come up with the book, I must admit. But I'm going to say that at this moment, it's a book that was written. It's not a large book, but written by Linda Lipson, and she is the curator of the herbarium at the BC Museum of Biodiversity, which, of course, I'm going to have a show there. And her book is called Pressed Plants, Making a Herbarium. And in actual fact, it is exceedingly useful for anybody who's interested at all in collecting and, pressing plants properly, keeping them like a real herbarium does, or even collecting for, like if you were collecting for the museum, she tells you about it. And I think that one of the things that immediately caught my interest is that she quotes from Robin Wall Kilmer, and it's about ethical collecting. Like things. Know the ways of the ones who take care of you so that you may take care of them. Introduce yourself. Be accountable as the one who comes asking for life. Ask permission before taking. Abide by the answer. Never be the first. Never take the last. It goes on like this, and I think that Linda is exceedingly sensitive, and I was thrilled to find that she would include that in her section on ethical collecting, because I think that's one of the more important aspects of collecting in these days when habitats are being destroyed, and here we are going out, just willy-nilly collecting things and not thinking about what we're doing as a result of that. Anyway, so Pressed Plants, Making a Herbarium by Linda Lipson.
And it has beautiful artwork as well.
Yes. And the other aspect, of course, of this book that is really wonderful is the fact that it's beautifully illustrated by Derek Tan. And he does these careful line drawings, which are easy to interpret. And, if you're much more visual than you are verbal, you'll get tons of information just by looking at his great illustrations.
Thank you so much for sharing both of your books. Recommendations resonate with this upcoming exhibition of the art, the plants, the intentionality, the relational aspects that I appreciate in both of you.
Thank you. And thank you very much, Kalika, for undertaking this and being a wonderful moderator.
Yes.
I think you're hired.
Fantastic. So, does that mean we get ice cream?
Yes.
Fantastic.
Thank you so much.
Thank you so much for listening. Show notes with links from each episode are available at the website pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's a form at the website where you can pop in and say hello and give me feedback. If you want to support the show, remember to leave a rating on iTunes, Spotify, or whatever podcast mothership you use. And finally, if you have the means and you want to help support my lab's effort to document bee biodiversity in Oregon, visit OregonBeeAtlas.org and follow down to the donate button where you can make a tax-deductible donation to the Jerry and Judith Paul Native Pollinator Endowment. Every little bit helps. See you next episode.